A very good evening to my wonderful champions out there. I missed all of your comments, all of your interaction, everything. And now I'm back here with the Sankalp series, my dear children. And I would love to start off this session with a very, very relatable question. Listen to me carefully. So how many times in your life have you felt that you have put in a lots of efforts, but then that was of no use? That is, ma'am, I studied for these many hours the whole day, but my marks were just this much. Well, I think it happens in everyone's life. At least it had happened in my life. Has it happened in your life? Let me know in the comment section. You know what? Mendel also faced the same thing. He worked on some experiment for 7 years of his life, my dear children. And later, people said that, hey, whatever you did is not correct. We cannot approve it. How would he feel? Obviously, he would feel sad. But then, is that the end of the story? No, my dear children. It would never be the end of the story. We all have to be patient because one day, just like how Mendel's works were rediscovered, were being taught as of now in schools, you are being taught about that. Similarly, in your life also, there will be a big up coming really soon. You just have to be patient for that. And with that ideology, we have to watch this session. I hope I have given you the initial boost of motivation. That is, you can still do it. If at all someone says you cannot, let them go. You believe yourself, you work hard, you will be able to conquer. So if you believe that, as I said, comment down in the comment section that, yes ma'am, I will be able to do it. Whatever you want to do, whatever is your affirmation, I will be able to top PU2, I will be able to crack KCET, whatever it is. I will be able to study for 5 hours a day or 3 hours a day. Whatever small affirmation you have, let me know in the comment section. And with that, let's go and talk about what's our plan. Okay, we need a plan, right? So over here we are at L3, level 3, where we are going to talk about dihybrid cross and chromosomal theory. So this class is going to be short and informative. I have seen that most of you children have doubts in dihybrid cross. So I'll take away all your doubts, my dear children, and I'll make sure that you are getting 13 marks in your PU2, and also you're grabbing all those 5 marks in your KCET examination. Well, in the next session, we'll be talking about linkage and recombination and also sex determination. Now, different mechanism. Okay, that session could be a bit longer. Now, L5 will be disorders in organism, where we will talk about sickle cell anemia, hemophilia, Down syndrome, all of it, with examples of people who actually suffer it. Okay, it could be celebrities, it could be anyone out there. So you will able to understand all those things better. And level 6 would be NEET plus KCET PYQ. So I hope you are ready, and let me thank everyone who commented down the comment section below. So we are yet to receive the the official handle. So once I receive it, I thought I would comment to you. I still can do that, but that the heart and sign and anything might not come. But just understand, if at all you see Jasmine Joy Deeksha, it's me who is commenting back to you. Okay, so yes, my dear children, thank you so much, whether it be Prajwal or Mansa who gave the answers like 1, 2, 3, 4, 5 in a very proper order. Kudos to you. Whether it's Sagar who gave me all the answers, I was actually astonished by the commitment that this child has. Okay, listening to each and everything and saying that, yes ma'am, I have tried a blood grouping thing and this was the blood group. I was really happy to see all that. Manasa saying challenge accepted, Prajwal saying challenge accepted, and I have a very cute comment from Rival Gaming who said that senior here got BMS College with only the help of Deeksha Karnataka faculty. We are so grateful to have you. Thank you so much for putting that efforts and commenting down in the comment section. So my dear children, now we are gonna begin with today's class. So all about Gregor Mendel's dihybrid cross experiment. First question that probably you might have to me is that, ma'am, what do you mean by dihybrid cross? So over here, I want you to imagine 2 plants. This is Pappu and that's Shappu. Okay, so over here, these 2 plants, I am going to take them into consideration, and I am going to talk about their 2 traits. One trait is the shape of the seed, another trait is the color of the seed. Shape of the seed and the color of the seed. So over here, the shape of the seed is round. As you can see, it is round, and the color, again, as you can see, it is what? Yellow. And over here, if you see the shape of the seed, it is what? Wrinkled. And and the color is, as you can see, it is what? Green. Now since I have two traits over here, shape and color, I am going to analyze that in their cross. I am going to cross them and see what happens to these two traits. How are they inherited together? Is there some change that I can observe in the next generation? This thing is known as a dihybrid cross, where I take two organisms and I consider the two traits in them, and I have to make sure that they differ in these two traits. So over here they are differing in the shape. One is round, other is wrinkled. One is green and other is what? Yellow. So I am taking this into consideration and going to check their cross. How will they be? So that is what you mean by dihybrid cross. So it's a genetic cross between two organisms that differ in two traits, to see how those traits are inherited together. Will I be able to see baby plants with seeds which are round and yellow? Will I be able to see baby plants with seeds wrinkled and green? Or is there something new that can pop up? So that's the question. Now over here, junior Mendels out there, are you ready for the experiment? I hope you are. So listen to me carefully. I took pure homozygous plant. That means they are homozygous, means, for suppose R stands for round, so it will be capital R itself, and they are yellow also, so it will be capital Y. I said homozygous, so it has to be the same. Now over here I am taking, so this is what? Round and yellow. This is round and yellow parent plant, and then I have one more parent plant, that is wrinkled and green. How do I show it? How do I show it? The recessive version of it. It will be what? Small r. Recessive version of y. It will be small y. Okay. And now, if we are talking about the chromosomes, how do they look and everything, for your understanding, I always make this, okay, it is going to help you really well as of now, but don't do this on the day of examination, okay? So over here, by this I mean that there are chromosomes where capital R gene is there. There are chromosomes where capital Y gene is there. Similarly over here, small r gene is there, and small y gene is there. Now whenever you are going to make the gametes, we have studied in law of segregation that the chromosomes, basically the factors that you say, they move away and only one factor comes into a gamete. So from this pair, only one guy can come in. So can I write capital R? From here also only one guy can come in, so it will be capital Y. How many combinations can you have? 4 of them. This can go with this, okay, this can go with this can go with this, and this can go with this. 4 gametes can be formed. So but over here, since all the gametes would be same, I am writing RY. So this is the gamete, capital R and capital Y. And over here, my dear children, I am going to take again, from this to this pair only one guy can come in, so it will be small r. From this pair only one guy can come in, and that will be small y. Again, how many of them would be formed? 4 of them. But since all of them will be same, I am just writing like this. Okay, now I want the F1 generation. F1 generation, what would that be? So it will be capital R, capital Y, small y. Ma'am, I didn't understand in the chromosome format, can you show? So over here, just think like this. So I got a chromosome capital R, a chromosome capital Y from this parent, a chromosome having small r and chromosome having small y from this parent. So this chromosome is going to come over here, so I am writing capital R. This chromosome is going to come over here, I am writing small r. This chromosome is going to come over here, capital Y. This chromosome is going to come over here, small y. So what do I have now? Capital R, capital Y, small y. This is what I mean by this particular thing. Now, this is the genotype. What is the phenotype over here? What is the physical appearance? Phenotype means physical appearance. How do they look? Are they round and yellow or wrinkled and green? So since capital R is dominant over small r, it will be round. And since capital Y is dominant over small y, it will be what? Yellow. So round and yellow, they are, or in F1 generation, all the plants that you see would be what? Round and yellow. Is this clear? This is Chutu thing. It will be clear to you. If at all it's not clear, look at here once more. Okay, it will be clear. Remember always, from a pair of chromosome, only one guy can come in. Okay, only one guy can come in. Okay, now over here, round and yellow, plants were being formed, seeds were there which had these round and yellow seeds. Plants were there which had round and yellow seeds. Now Mendel was like, I did the same thing in the monohybrid cross. I want to cross these plants. within themselves, among themselves, that is among the siblings there was crossing being done. Among the progeny of F1 generation, again a crossing was done. Now for that, this would be the parent, right? The next cross, if I am doing, my dear children, what will happen? This would be the parent, capital R, small r, capital Y, small y. Similarly over here, capital R, small r, capital Y, small y. Now you tell me what would be the gametes. From this pair, one guy can come in, capital R suppose. From this pair, one guy can come in, so suppose capital Y. Similarly, capital R, small y. Agreed? Then small r, capital Y. Then small r, small y. So how many type of gametes? 4. But this time all of the 4 are what? Different in nature. If at all you were not able to understand this, listen to me. So we had something, okay, there's no space over here. Just a second. Can I make it here? I hope we can. Yes? So children, this is the scenario. Capital R, small r, capital Y, small y. Agreed? This guy can go with this. From a pair only one can come in, so capital R, suppose it came in. Over here capital Y can come in. Now in the next time, next gamete, this and this can come together, capital R and small y. In the next gamete, small r and capital Y can come together. In the next gamete, small r and small y can also come together. Similarly over here also, you have to plan accordingly. Same thing would happen, same type of gametes, capital R, capital Y, capital R, small y. You don't have to remember it, don't mug it up, okay? Both of them are dominant. Here one is dominant, other is recessive. So capital R, capital Y, capital R, small y. Similarly, small r, capital Y and small r, small y. Easy. It's easy. Okay, do not get too much confused. So we should have capital R with capital Y, capital R with small y. Similarly, we should have small r with capital Y and small r with small y. Now these are the gametes here also. How do you cross it? Ma'am, if I keep on crossing like this, I'll be going all mad. So, which is why you should be using a Punnett square over here. Who gave the Punnett square? The question number 1, homework question number 1. Who was the person who discovered Punnett square? Was he from British origin? Was he from German origin? What was his origin? You have to tell me that also. Now, if at all you are preparing for NEET, or if at all after preparing for biology you think that I should be giving NEET once, this question could come there. So, please tell me homework number 1, who gave the Punnett square? Okay, was he from British origin or German origin or what origin? You have to tell me that as well. So my dear children, what he did was, Mendel basically with the help of Punnett square, over here, not Mendel with the help of Punnett square, basically just us, okay, us with the help of Punnett square, we can find the answers very easily. So we put all the gametes in one side, again all the gametes in one side, as you can see over here. And now I am going to cross, okay, and now I am going to cross, let's cross. So suppose capital R, capital Y gamete, and capital R, capital Y gamete came in, so it will be capital R, capital Y, capital Y. Similarly, this and this came in, this would be the thing. So one capital Y and the small y. Again, you have to cross this with this with this, and that's how you make the whole thing. How many progenies would be produced, my dear children? 16 of them. 4 here can combine in whatever ways, and 16 of the progenies would be produced. But you know what was the unique thing about it? The unique part of the story is that, my dear children, out of 16, out of 16, everything went out. Okay, out of 16 which was there, 9 of them were showing the dominant trait. Ma'am, what is a dominant trait? Remember round and yellow? Round and yellow was a dominant trait, no? So round and yellow. 9 of them were showing that trait. 1 of them was showing the recessive trait. What is a recessive trait, my dear children? Wrinkled and green. Wrinkled and green. Now, there were 3 of them who showed a new combination altogether. Round, not with yellow but with green. So, round with green. Again, next 3 of them which showed wrinkled, not with green but with yellow. So, that was the new combination. Non-parental combination is what we call it. That means it was not found in the parents, it is just found in the progeny. So what is this? Non-parental combination. How many of them? 3 plus 3, 6 of them were very, very new, my dear children. And this ratio which you see over here, that is 9:3:3:1, is the phenotypic ratio. The physical appearance. I am not talking about genotypic ratio, I am talking about the physical appearance. So this is the phenotypic ratio of what? Of F2 generation of dihybrid cross. And with this, what do you understand? Why did Mendel do all this experiment? Was it of some use? The answer to this question lies over here. Did Gregor Mendel find something unique? Yes. The law of independent assortment. Ma'am, big term, I don't understand. It's very easy. Okay, this term is very easy. So what is this law about? So I have made two hands like round and yellow. So do you think they're always together? When we saw the, when we saw the cross, did you always found that round was with yellow? No, right? 9 of them were round and yellow, but 3 of them were what? Round and green. Okay, round and green. With that, what do we understand? We understand one very important fact, and you have to listen to me very carefully. Okay, so over here, if this were the chromosomes, here capital R, small r is there, capital Y, small y is there. Okay, whenever gametes were formed, it was not like capital R will only go with capital Y. Was it like that? Was it like that? Capital R will only go with capital Y? No, my dear children. This was a combination, but capital R could also go with small y. It also could go with small y. So that means they are not dependent on each other. They are independently getting separated. This and this guy is independently, this and this guy is independent. So over here, according to law of, when you say that this and this is getting separated, that is law of segregation. Law of segregation that says that when a pair of factor is there, both of them segregate and go into each gamete. Now when you're talking about one trait, genes of one trait and the genes of another trait, we are saying that they also independently assort from each other. Capital R has nothing to do with capital Y. So there is no connection like this. Again, I'll repeat. When two pairs of traits are combined in a hybrid, when two pairs of traits, okay, this is one pair, this is another pair. Segregation of one pair of character is independent of another pair of character. This has nothing to do with what is happening over here. Okay, they are different. What is it? Segregation, separation. Segregation means separation of one pair of character. This is one pair of character is independent of what? Segregation of another pair of character. This is law of independent assortment. Now children, get confused with law of segregation. Law of segregation in that only you're talking about one pair, not two pairs. Okay, here you're talking about two pairs and that they have nothing to do with each other. This is law of independent assortment, which was put after doing the dihybrid cross experiment. I hope this is clear, which is why in the progeny generation it was not always round and yellow. It was also round and green, and it was not always wrinkled and green, but it was also wrinkled and yellow. You were able to find new combination because of this, because of the law of independent assortment. Please understand this. I hope this is clear, children. Okay, so that's all about this particular thing. Now, but what, is there something that I'm missing? Okay, but what went wrong? So I told that he did this, it was really good, and that's why we are able to find that new characters can combine, and that is happening in our life also, no? We find that all the new characters are combined, and that's how me and my parent are not exactly same, we are different because of so reasons. Now, what went wrong when all of this was actually correct? First thing that went wrong in his life was communication was difficult. So his work wasn't widely known at that particular point of time. Calling, emailing, things that were not there. So which is why communication was difficult. That I have found something. Whom will I call and say? How can I call and say? That was one problem. Next thing, his idea of discrete factors or no blending was not accepted. So he said that tall and dwarf is the only feature. There is no medium heighted thing. There is no blending. that was not accepted. Okay, so his idea of discrete factors, that is unique factors or genes, my dear children, and no blending was not at all accepted. Now third thing, he used mathematics and biology. That was very unusual. No one believed that. For them, people, my dear children, biology was biology. You cannot apply mathematics in that. That was the scenario up there. So they were not accepting all of this thing. Do you know that he was actually a physics enthusiast, and then he slowly diverted his things into what? Biology. So obviously he had some tactics around the logical things, around the mathematical tools. So he used that to explain all these terms, but no one actually accepted it. Okay. Now fourth thing, he had no physical proof of what? Not genes, factors were or how they looked. He just said that this probably could be happening. He never could explain that how do they physically appear, where were they actually located, nothing. So in that case, you only tell me. If at all you come to me and tell that, ma'am, this thing happened, this thing happened, obviously, or basically I come to you and tell that, hey, you know what, Prajwal, hey, you know what, Manasa or Sagar or anyone else out there, that this is how things happened. Will you believe me if I do not give you a proof? No. If I teach you something else, okay, very new, without a proof, will you believe me? No. That's very difficult. So similarly over here also, we needed physical proof which was not there, and that's where he went wrong. So these were the four things where he actually was not able to establish himself, and which is why there was a very big downfall in his life, just like how we have in ours. So all his efforts were going waste, my dear children. But then, okay, there's a homework question. Before I go ahead with the story, there's a homework question for you guys. So in the F2 generation of Mendelian dihybrid cross, the number of phenotypes and genotypes are. In the F2 generation, how many phenotypes were you able to observe? How many phenotypes were you able to observe? 4, no? Round yellow, round green, and wrinkled green and wrinkled yellow. That is what you found out. Now, how many genotypes were there? So don't tell me 16 genotypes were there. If you tell me 16 genotypes were there, I will jump from this block to that block. That's all. Nothing would happen to me. I was chumma kidding. I won't go anywhere, okay? So over here, my dear children, please understand this fact that if you're asked about number of genotypes, like 1, 2, 3, 4, 5, 6, 7, 8, no. There are things that are getting repeated also. You have to check that. Okay, I will give you proof. If you don't believe me, I'll give you a proof. So tell me, this and this looks the same, right? This also looks the same. So don't you think the genotypes are repeated? Yes. So you have to do that homework and tell me, count. What are the unique genotypes which are not repeated? Okay. The unique genotype. For example, this thing, I will count it as just 1. Okay. That is unique. That is only 1. So you have to find it out and you have to tell me in the comment section below. Will you be doing that? Will you be doing that? I hope you do that and let me know. Okay. So that is one homework for you. This is homework number 2. Homework number 1 was Punnett square. So please take it seriously, my dear children. You do not have your unit test coming along now. You have your midterms, and in midterms we want full on full, right? So make sure you're doing that. And I'm telling you, for all the competitive level examination, for at all you're looking for some scholarship or things like that, you will need a minimum percentage requirement. So make sure you are achieving that. You should be overachieving that. Okay? So that is one thing. Now going ahead, okay, so I was talking about this thing. That is, there was so much downfall in his life, but then now came a twist in his life. Is it a good twist or bad twist? It's a good twist. But the problem is that it came after 37 years. Okay, after Mendel passed away, that's when this particular thing came up. What particular thing came up? Rediscovery of Mendel's work, my dear children. So there were 3 scientists who said that guy you said who was wrong, my dear children, he was not actually wrong. He was actually correct in so many senses. Yeah, there were certain deviations from what he said, but that does not mean he was wrong. So my dear children, in the year 1900, so remember it's not 1990. Okay, children, do that thing. It's not 1990, it was 1900. Where 3 scientists came in together. You have to remember their names. One is Carl Scorens, the other is Eric von Schemmark, and the third one is Hugo de Vries. So in your NCERT, it's written like de Vries, okay, Corens and Schemmark. The T is silent, okay, Schemmark. So these three people in the year 1900 came back, rediscovered Mendel's work 37 years later. How were they able to rediscover Mendel's work? They found out all the documents and stuff like that, and they had better tools. By this particular time, my dear children, Mendel did his experiment from 1856 to 1863. Now after so many years, 37 years, so many things change. You only think from this point onwards, or 5 years back, was everything the same? No, right? So obviously after 37 years, a lot of things changed. Better microscopes were available. Okay, and during this time, they revealed the chromosomes structure as well. The chromosome was visible. So with the help of staining, okay, they were able to find that there were certain colored bodies. Those were known as chromosomes. Where were those things? In the nucleus. So it was also found that they doubled, okay, they duplicated, and they were split during what? Cell division. So just like how Mendel said that factors, they split, they move to each gametes. So basically what are we trying to understand from this is that he never knew that there were chromosomes. He never knew there were chromosomes. He just said that there were factors, and factors segregated. So don't you think from your end that these factors probably might be on chromosomes, and whenever a chromosome separate, my dear children, the factors were also separating? Yes. So this was found out, not the other part of the story, but at least that the chromosomes were there, and the chromosomes doubled, that, and also that they were split. Now on the basis of this information that the chromosomes were doubling and splitting and things like that, two more people came into the picture, and those were whom? Walter Sutton and Theodore Boveri. Very, very important. Okay, Walter Sutton and Theodore Boveri. So they came into the picture in 1902, just 2 years after these three guys came in. Okay, and said that, yes, you are correct. You just rediscovered Mendel's work, and I totally agree with what Mendel said, that factors were separating. So they observed that chromosomes were separating, and probably these chromosomes had those factors that Mendel was talking about. So this is a time when these guys came in and said that, yes, this probably could be the scenario. So 1902, chromosome movement during meiosis had been worked out properly. Okay, during meiosis that the chromosomes were moving and this and that. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes, and used chromosome. Okay, so they did what, my dear children? The behavior of chromosomes was parallel to the behavior of genes, or basically what? The factors. That means however the chromosomes were moving, similarly, in the same pattern, something was being followed up, and that is the function of the genes. They were also moving according to that. I hope you got it. Okay, so please remember this. The behavior of chromosomes was parallel as in same, was parallel to the behavior of what? Genes. That whenever genes are segregating, chromosomes are also getting segregating. So basically, probably it's because of the chromosomes. Whenever they segregate, the genes on them also segregate. And that is how they gave out the chromosomal theory of inheritance. So both of these guys, they studied very, very enthusiastically about this and gave out the chromosomal theory of inheritance. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors that they carried. Okay, so whenever there is pairing of chromosome, they saw that factors are coming together. Whenever there was separation of chromosome, they found that the factors were moving away from each other. So with that, they gave the chromosomal theory of inheritance. Okay, this is the chromosomal theory of inheritance. Pairing and separation of a pair of chromosomes would lead to what? Pairing and segregation of a pair of factors that they carried. It was not genes, it was factors. Okay, and now Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. So if in your option it is given that who gave chromosomal theory of inheritance, if you have an option like Sutton alone, my dear, you can go with that, but actually both of them worked together. But in your NCERT, one line is mentioned that Sutton was the one who understood that yes, chromosome separation is there, and he mixed it with Mendel's theory also, and then he found that okay, he was talking about factors, and that is also getting segregated. So with all of this information put together, he gave the chromosomal theory of inheritance, which said this, which was this. Okay, so that's about the story. Now obviously again, whenever someone says something like that, you again need to prove that. And who proved that? Who proved the chromosomal theory of inheritance, my dear children? It was Thomas Hunt Morgan. It was Thomas Hunt Morgan who proved the what do you call chromosomal theory of inheritance, and that is what we are going to study in the next session. Again, you are going to see twists and turns and things like that, but I am pretty sure it will be very, very useful for you guys. Okay, done? Deal? Now, going ahead, I have a question that I would love to solve for you guys, because I think it's kind of confusing for some children out there. So there was a question in KCET, a pure breeding pea plant with round and yellow seeds, just like how we did, okay, was crossed with a pea plant having wrinkled and green seeds, okay. On selfing of F1 hybrid of this cross, 64 progenies were obtained in the F2 generation. So F1 was crossed, and you got 64 progenies, my dear children. Find out the number of F2 progenies showing the non-parental character. So you know one thing, that is the total number of offspring is what? 64. And now what do you have to find out? Out of the 64, how many of them showed the non-parental character? What do you mean by non-parental? That is the ones which is not there in the parents. For example, it should not be round and yellow, it should not be wrinkled and green. So what's left? Round and green and wrinkled and yellow. Right, that is a new combination. So how do I find out? So we know, according to Mendel's dihybrid cross, there was a ratio 9:3:3:1. Out of this 3 plus 3 of them were what? The non-parental ones. Were what? The non-parental ones. So basically, and we also know that basically out all of them put together, there were 16 of the progenies which were produced. So 6 out of 16, my dear children, were the non-parental. Now you have to find the same for the 64. So what do? You do like this. Okay, you do like this and then you find out. So basically over here, if I 16 into 2 is what? 16 into 2 is 64. Oh sorry, 16 into 2, wait, 16 into 4, not 16 into 2. 16 into 4 is 64, 6 into 4 is what? 24. So 24 of them were what? The non-parental ones. I hope you understood. So 9:3:3:1 ratio. In that, 6 of them out of 16, my dear children, they were the non-parental ones. So if you want to find out of 64, how many of them will be there which would be having non-parental traits? What do? 6 divided by 16 into what? 64. So you will get your answer. I hope that's clear. Okay. So your answer would be 24. Now, if at all some questions comes like this, will you be able to answer it? I hope you would be able to. Now, a homework for you guys. Now, this thing you can do, which is why I have kept it as a homework. This is homework number 3. In a dihybrid cross in pea plants, Mendel got 9:3:3:1 phenotypic ratio. It denotes that. What does it denote? When you talk about Mendel's dihybrid cross, which had this ratio, what does it denote? That the alleles of 2 genes are interacting with each other. The alleles of 2 genes, that is a gene for shape and the gene for color, are they interacting with each other? It is a polygenic inheritance. We studied polygyny in the previous session. If at all you do not know, please go and see it. Polygyny is where you have multiple genes coding for the same trait. No, you have just one gene, right? One gene, that is capital R or small r, coding for what? The shape, right? And capital Y, small y, coding for the color. So that is not a polygenic inheritance. Now it is multiple allelic inheritance again. Okay, for one trait, do you have multiple alleles? Just 2 of them, no? Capital R, small r. For another trait, you have capital Y, small y, that's all. Now the alleles of 2 genes are segregating independently. Now do you tell me the answer? So I basically explained all of it. You can actually tell me the answer in the comment section below. If at all you find that I'm not able to understand this, you can ask me that as well. Okay, ma'am, can you explain that? I will explain that. Okay, so in the next session, along with your winners of the homework quiz out there, I'll be talking about the answers for these questions. So if at all you're facing any trouble, make sure you're watching that video as well. Is that clear? I hope that's clear. And my dearest children, lots of love to all of you. I hope you're studying really hard. Okay. So, like Gregor Mendel, we would also have ups and downs in life. That does not mean that we should be disappointed. We should be working really, really hard for it. Gregor Mendel did that. He worked hard until the end of his life. Okay, so we should be working hard, and whenever we will work hard, we will be rewarded for it. Wishing you all the best. Lots of love from Jasmine Ma'am. Take care of yourselves. Bye-bye. And in advance, Happy Independence Day.